Sorry, now, so now we have live. So just wait a second. Stop sharing for a moment, Martina. Yes.
Okay, let me stop sharing.
Yes, just for a second. Just this
Okay,
is
okay.
Thank you,
Okay.
thank you, Cyril. No, because I was connecting the Zoom with the YouTube. I forgot to go live in YouTube. So now these are the things. So welcome everyone again. So I won't say again the same thing. So welcome everyone. This is the 2020 annual meeting from ARRIGE, this time remotely. And without further ado, we just can start with the first presentation. This is a keynote lecture we have from Martina Crispo. She is responsible for the laboratory where they produce and modify mammalian embryos of different species. And she's working at the Pasteur Institute in Montevideo, Uruguay. And we're glad to have you here, Martina. So please, you can start sharing your presentation. Thank you. You have 30 minutes, out of which 20 for the presentation, and the rest will be for the discussion. Thank you, Martina.
Okay, thank you. I will try to share the screen now. I think now I'm sharing, and thank you for inviting me to give this keynote lecture at the ARRIGE annual meeting. I hope you hear me well. And yes. I tried to make a simple presentation of all the incredible advances we have heard over the last year on gene editing field. So let me say that it was a huge amount of work because there are many great papers in this last year, and I will try to explain in simple words this, and at the last minute we can discuss and we can exchange further for the ethical point of view. So let me switch here. Okay, let's see if it works. Yes. And as Luis said in the General Assembly, I cannot start this presentation without acknowledging Jennifer Doudna and Emmanuel Charpentier for awarding this year the 2020 Chemistry Nobel Prize for the development of a method for genome editing. And this was great news for our field. We were all very happy, although there were some researchers that were leave out of this prize, but I think that always happens in the Nobel Prize, so we are very happy to receive this prize for our field. And this is just a reminder of how CRISPR works, and this is the basic work of CRISPR, because now it has changed a lot. But basically, the classical approach of CRISPR is the break in the double-strand DNA. And this break can be repaired by endogenous repair mechanism of the cell, by non-homologous enjoining, creating a knockout, or by homologous direct repair, creating a knock-in. And also, CRISPR can mutate RNA, not only DNA. So this was described a few years ago and is the same approach. But CRISPR, you only need to add some PAMRs, and CRISPR can also mutate single-strand RNAs in the same way. And this is coming more and more complicated nowadays because the structure of CRISPR is changing, and they are discovering new CRISPR systems. So I'm going to explain in this lecture about this. What are the applications of gene editing? There are several applications. One of the most widely spread applications is the generation of animal models. In this case, I work with the generation of animal models, mostly mice, but also large animal models. There are hundreds of papers coming out using this technology to generate, for example, cancer or neurodegenerative disease animal models, models of infertility, several models of human or animal disease, also models for enhance animal productivity when you work with livestock species. And let me show you our last large animal model that was published this year in Scientific Report. This is a sheep we have generated in collaboration with the group here in Uruguay and in France. that in which we mutate with gene editing the autoferlin gene. The autoferlin gene is a gene that appears mutated in humans and impairs the audition of humans. So we have generated this large animal model to test different therapies for recover audition in humans. So for that, we have transferred around 1,300 embryos, and we have obtained good editing rates. Around 18% of the lambs born were edited, and from this 18%, we have obtained around 62% of lambs with the mutation we wanted. And so these chips are available to test different therapies against deafness. What else? What can we do with CRISPR? We can use them for human immunotherapies. This is a field that is growing a lot in the last year. In this case, for example, this is an article that has recently appeared from Liu et al. And they edited T cells, normal T cells from patients with refractory non-small cell lung cancer. In this case, they retrieve the cells from the patients, and with using the classical approach of CRISPR ex vivo, they electroporate the Cas9 and the single guide RNA to mutate the PD-1 protein. PD-1 protein, when you mutate this protein, you increase the immune reaction. The PD-1 is an immune checkpoint. So they put again the cells in the patient. This is a clinical trial in 12 patients that receive again the transfusion of these mutated cells, and they were monitored for up to 96 weeks, and they found no severe treatment-related adverse effects in this case, and very low off-target editing events. And that accounts and start to add more information for the immunotherapies using CRISPR. Another approach that has recently came this year using the same approach with CRISPR. In this case, this group edits also ex vivo stem cells from a patient with HIV, and they mutate the CCR5 receptor, and they put the cells back in the patient. There was only a single patient. In this case, in this other paper, they used three patients with refractory cancer, and they use this therapy as a composite therapy, and they do the same approach. They retrieve the T cells and they mutate the T cells ex vivo for the immune checkpoint also, and they put the cells back. And they found in all these papers that the efficiency varies significantly across target genes and patients. It depends on the gene where you want to mutate, is the efficiency you find. In these two papers, for example, the mutation rate was about from 5 to 20% of the cells had the edition they wanted. And there is a need to evaluate the long-term safety of CRISPR. Of course, these are very short reports, clinical trials. They found that this technology is rather inefficient, and they say they are ready to test base editing and prime editing. So we already heard about base editing some years ago from some groups working in this, where you can specifically mutate one single base pair in the DNA. And now lately we have heard about prime editing. So what is prime editing? Prime editing is a very sophisticated tool that has recently appeared from the group of David Liu. This was published in October last year. And in this case, they have managed to use an impaired Cas9 fused to a reverse transcriptase, okay, and a prime editing guide RNA called pegRNA. Here you can see the tool, okay, with the Cas9 fused to the reverse transcriptase and the pegRNA. And with this very, very precise tool, they can manage to do targeted insertion, deletion, and they can arrange 12 types of base substitution without making double-strand breaks in the DNA or without introducing donor DNA templates. So this is something that is improving a lot the field of genome editing. And they found higher or similar efficiency and fewer byproducts than homologous direct repair, and complementary strengths and weakness compared to base editing. So, and they didn't find many off-target editing places, much lower off-target than Cas9 nuclease. With this, they proclaimed that it's possible to correct up to 89% of known disease-causing gene mutations that are due to a single base pair mutation. So, with prime editing this year also, recently in October, this group has managed to mutate cells. They obtained cells from patients with several diseases like Wilson disease or patients that had deficiency in the DGAT1 gene. And in this case, they obtained these cells and prepare in vitro organoid culture models. And in this case, they recover the disease-causing mutation in these intestinal or liver organoids using the prime editing with greater precision than homologous direct repair. So they also compare in the same paper base editing against prime editing, and they find that base editing is more reliable than prime editing. That means there is a lot of work to do also with prime editing because it's very recent, this technology, and they didn't find any off-target effects. So this is something new, this is working, and we have to wait for more information regarding this prime editing tool, but it seems that it's getting more and more, the gene editing field is getting more and more complicated. So we have base editing, we have prime editing, and we also have dual base editing. What is dual base editing? Based on the prime editing, on the base editing that was originally published a few years ago, now some researchers working in plants, maybe we will talk about that later, managed to fuse this 2 deaminase with Cas9 nickase and make the two editing in the same target site. That means they convert CT and AG on the same target time, not only one editing. And they found less adenine activity, more cytidine activity, and less off-target effect than base editing. And lately, in June, there were several reports in the same number of Nature Biotechnology, like 3 reports using this dual base editor that seems to work also in human cells. So we can mutate DNA, we can mutate RNA, and we can also mutate mitochondrial RNA. So you can see this is getting more and more complicated and sophisticated. This year, in May this year also, the group of David Liu, he's a very active researcher in this field, has published the possibility to mutate mitochondrial RNA using base editing. In this case, these researchers use a toxin from a bacteria called DDDA. This toxin, they split the toxin in half, okay? So in order that not let them be toxic, and they fuse it to a TALEN and an uracil glycosylase inhibitor. So in this case, they managed to assemble this RNA-free DDDA derived cytosine-based editors. Okay, and with these base editors, you can catalyze CG to TA in the mitochondrial RNA. And they found that this high target specificity and precise mitochondrial RNA manipulation. In cells, they found that this makes some changes in respiratory rates and oxidative phosphorylation. And this can have broad implications to study and potentially treat mitochondrial disorders that are some very complicated disease to treat, because until now there were only possibility to destroy the whole mitochondria with the consequence that can have. So now we have this new tool that can aid to avoid these mitochondrial disorders. And again, for editing RNA, another system that has recently published uses another approach called ADAR. ADAR accounts for adenosine deaminase acting on RNA. And with this new tool, those are human proteins that can mutate double-strand RNA. With this new tool, we can change in the double-strand adenosine for inosine. Or if you use this other approach, ApoVC, we can mutate C. We can mutate the C also. So these proteins does not generate, at least they said, no immune reaction in the patient because they come from human. They are human proteins. And with this, they proclaim that they can correct human diseases like muscular dystrophy, cancer, acute pain, cholesterol levels at the RNA level with high efficiency and high specificity. They also state in the paper that the regulation of this therapy should be easier because there are many RNA or already approved drugs in the market. So as you are going to mutate only RNA, the regulation should be easier. And amazingly, another discovery from this year from the group of Jennifer Doudna is a new CRISPR system found in huge phage. Phage are virus that infect bacteria, and these huge phage from around 500 kilobase have another system of CRISPR system called CRISPR-Phi. Let's call it CRISPR-Phi. And they found that it is a hyper-compact genome editor. You can see it here in the upper. I hope you can see the mouse. You can see it. It's very small. It doesn't have the Cas protein genes, and it's half the size of, for example, Cas9 or Cas13. So it's very small. It accounts for a 33% of cell addition in this paper. And we still need a lot of information about that because we don't know if it's more specific than previous CRISPR-Cas system. Is better for in vivo therapy? Maybe yes, because it's smaller and we can deliver it easier to the patient. And finally, we have to take into account CRISPR for this new pandemic, COVID-19, that we are all aware of. Does CRISPR work for COVID-19? I have to say yes. I have been reading lately a lot of papers working against COVID-19. In this case, there are several approaches that have been used before for DNA or RNA virus, like Sherlock from the group of Fen Sang, or Detector from the group of Jennifer Doudna. In this case, they use Cas13a or Cas12 for another virus like MPV or dengue or Zika. This method seems to be rapid, inexpensive, simple, and one of the advantages of this method is that no equipment is needed, no expensive equipment is needed. So the idea is to have this method anywhere of the world, and that anybody can do it. So how do they work? I will try to explain the rationale on this method. In this case, we have the Cas, for example, for the method of Sherlock, we have the Cas13a enzyme linked to the spacer and the target, and this enzyme will cut the viral RNA in a specific precise site, but after it cuts the RNA from the virus, it will recognize several similar sequences around, and it will very, very precisely and efficiently start to cut all the similar RNAs, and in this case they add a fluorescent sensor, RNA sensor, linked to a repressor in the same system. So the enzyme will cut the RNA from the virus and will cut also this RNA that is similar to the virus and will release this fluorescent dye, and this will, by colorimetry, we can detect then the RNA from the virus. And detector is from the group of Dona. It's very similar. It has only a previous step that we have to take into account because it uses Cas12, and Cas12 cuts DNA. So in this case, we have to convert RNA with a reverse transcriptase to RNA, and then to a DNA, sorry, and then it works in the same way with this fluorescent sensor. In May this year, the FDA approves a rapid COVID-19 test that uses CRISPR, the test from Fensang, as an emergency test, and this has been called Stop COVID, and it's very simple to use. You have to take the swab, the sample, or the saliva sample, and then you put the sample into the Sherlock system, and the only equipment you need is this heat Sherlock that will heat for 60 minutes at 60 degrees, and then you just dip a strip into the system, and you can check if you have one or two guides here. If you have two, you will see that it's positive. If you have one, you will see that it's negative. And lately we have detected that we have heard about an approach from Jennifer Doudna that accounts for a new test for coronavirus that can detect coronavirus in just 5 minutes. This is very new, and this uses Cas13a using a mobile phone, and we will see if it works, but it would be great if it works. So for treatment, CRISPR can also work for treatment. In this case, there is a system called Pacman, and Pacman can detect RNA from the virus very efficiently using CRISPR-based strategy. They will produce inhibition and degradation using Cas13d. There are a lot of acronyms for this system. Pacman is one of them, and they tested in vitro with a virus, and it was very, very efficient also. With 6 CRISPR RNAs, you can target more than 90% of all coronavirus. But we still have ethical concerns. This is very recently reported. I'll look at the date, October 31, because there were some researchers that report some on-target edits, large-scale and intended DNA deletions and rearrangements in the area surrounding the targeted sequence in human embryos. So we need a lot of information regarding these technologies, because we can find also these on-target edits that can be very, very detrimental. As concluding remarks, let me tell you that this gene editing system has versatility. There is a new therapeutic paradigm targeting diseases at the RNA level. We have to take into account delivery, administration, safety, preclinical study design, drug manufacturing, and economic and regulatory issues. But we still have a long road ahead. So with that, let me say gracias, and I'm ready to discuss with you further and exchange the ideas about these novel technologies. Thank you very much.
Thank you very much, Martina. You can stop sharing your presentation, and so we can, because there's a couple of questions already arrived here. So without further delay, I will ask you the questions. So Guillaume Levrier is asking, why did you choose to work with lambs to do animal models? Is it easier technically to do than macaques?
Yes, I think yes. We have any facility here for macaques. It's nice, but we have a strong collaboration with a group of researchers here that work with animal reproduction, mostly in sheep, and that's why we do this collaboration with them, and so we produce with them the embryos in vitro, and we mutate them. The system is really similar than mice, and then it's more complicated, the embryo transfer and all the things that this group does outside.
Thank you. Then there is another question from Guillaume, says basically it is due to institutional processes, but I guess
Hello?
you already answered this. Then we have Shalini Reddy, and she's asking regarding prime editing. Won't the reverse transcriptase in prime editing cause non-specific activity?
Well, I don't know, because this is very, very new, and the idea is that it's very specific of the target site you want to edit. I hope it does not make any other edit, but we still, as I said before, we still need more information regarding these new technologies, and maybe somebody there can ask more properly that is using prime editing. But as I said, it's very new.
There are few cases in which prime editing has been used outside the David Liu's lab. So we need to pay attention still. So there is another more specific question from Shalini Reddy on prime editing, but then we can skip this and we can go to Guillaume Levrier. He has another question regarding the diagnostic tools, and he's asking whether any of these Cas-based diagnostic kits are available in the market.
The only one I know is the one from Fengsang that is called for COVID. You are asking, right?
Yes.
This is in a small amount of patients, and this is, but it's not ready to be used for everybody. And it seems to work.
They seem to have an FDA emergency approval, but nothing. It's at least in Europe, no one of these different kits has been authorized.
No, so, we have a lamp approach here that can give you results in 1 hour, but also it's not for everybody. So they managed to have it for only a few cases.
Okay, then since we don't have more questions, we can go back to the question I skipped. So that's Shalini Reddy again, and he's asking regarding prime editing, whether because of the long RNA is also binding to the protospacer, whether this might have some implications in reduced efficiency.
Sorry, can you repeat the question, yes, Luis, because I didn't understand if the long RNA.
No, because we're having some difficulties with the connection, Martina, and but I'm repeating. So basically, for the prime editing, what she's asking, whether because this long RNA is binding to the protospacer, whether this might have some implications in reducing the efficiency.
I really don't know about that, but maybe yes, can have reduced the efficiency, although I think that there are still a lot of field to work in this prime editing new technology to improve the efficiency, because they say base editing is more efficient than prime editing, but maybe yes, you're right.
I think I have a question. What you have presented, and I do entirely agree, it looks like the base editors are the next big thing. So base editors in all the different varieties will probably be replacing most of the standard uses with traditional CRISPR-Cas, because the efficiency is being increased and the specificity is also being increased. Do you think it's the time for base editors? Martina.
Sorry, the connection is very bad and I have problems with that, but I think, yes, base editing will replace the standard approach. We would like to try it very soon in our animal models because they seem to be much more efficient. And as I see during these 2 weeks that I have been reading information, it's getting more and more sophisticated and more efficient. So it will be maybe the future for that.
Thank you. So there is yet another question from Prime Editing, a lot of interest in Prime Editing, and they say whether this has been.
I imagine. I'm not an expert in Prime Editing, but
oh, yes,
I read and there's not many information.
No, let's discuss whatever the discussion. The question is regarding whether Prime Editing has been used on human embryos.
Not that I know, and maybe we will have more information soon. But the only reports I found was that the first one and this one in human organoids from liver and from intestine, but not in human embryos.
Thank you. Well, we will have to finish. So muchas gracias, Martina. Thank you very much for your nice presentation. I think it was a good start for this meeting. So now let's move now to the next session. Now you can disconnect your camera. Thank you. And we can have Virginie. Virginie, bonjour. How are you?
Hello. I'm good, thank you.
Okay, so you're going to be sharing Nick. So Nick is here. Hello, how are you?
Hello.
So again, you will have one presentation by Nick, 15 minutes, and then it will be Gaetan video that will be 15 minutes, and then we will have 10 minutes discussion. So Virginie, the floor is yours.
Okay, thank you. We already done half of the job, but thank you, and good afternoon, morning, and evening from wherever you are. So I'm very pleased to introduce the speakers of the next session. So our first speaker is Nick Mead, who is Director of Policy at Genetic Alliance UK, which is the national charity working for genetic, rare and undiagnosed conditions in the UK. It's an alliance of around 200 patient organizations, and Nick leads the work which focuses on the commissioning of healthcare services and access to therapies on genetic testing, genomic technologies, and on reproductive choice. So Nick will present to us the work done with Genetic Alliance patient community on their views of genome editing.
Thank you, Nick. You can present.
Thanks very much, Virginie, and thanks very much for the invitation to speak here today. Virginie's saved me a second or two on describing the organization I work for. We have more than 200 patient organization members in the UK supporting people living with rare and genetic conditions, and we run the Rare Disease UK campaign, which works closely with Eurodis. I want to start just by acknowledging all the people that have helped us with our work on genome editing. Certainly all the people living with rare and genetic conditions that have contributed to our work, but also to Virginie herself for working with us on our policy work in this area, and to the Progress Educational Trust to collaborate with us closely on this topic. So today I'm aiming to share a little bit more about our community, talk about unmet health need in that community and how that affects our attitudes to medical innovation and therefore genome editing. I'm going to talk a bit about findings from two of our projects that have engaged our community in the topic of genome editing. And then I'm going to reflect a little bit on learnings from broader policy work that our organization has done that might reflect back on genome editing. So despite the political environment, the UK still follows the EU definition of a rare condition. affecting fewer than 5 in 10,000 people. And almost every single gene condition is rare. So the OrphaNet database shows us how many rare conditions there are that have been named and understood. That number's over 6,000 now. Around 70% are genetic and around 70% are exclusively pediatric in their onset. There's an amazing spectrum of in all types of attribute to rare conditions and how they bring challenges to the people they affect. But there are some patterns. One is certainly around how difficult it is to get a diagnosis, but also, and most pertinent to our topic today, is that there are very few treatments for people with rare conditions. Treatment at the moment for most conditions is trying to limit the progress of the condition. And then there's lots of other challenges that our community face, and they bring their own challenges around working, learning, and some of those challenges lead to mental health challenges too. So just to expand a little bit more about unmet health need here. So this is a graph from the ARRIGE, who have been trying to amplify research and progress towards new medicines for rare conditions. They have this graph that charts new medicines since 2010. These numbers are cumulative, and you can see that we've gone from very few to almost 400. So these are indications, not medicines, and that it combines Europe and America's regulatory figures. Just as an indication of progress, I know it's an artifact, but it's interesting to note that in this same time period, the number of rare conditions that we've understood has grown faster than the number of medicines we've managed to gather. And of course, there are other treatments that aren't medical, such as bone marrow transplantation, and adjusting diet to cope with metabolic conditions. But for most of these medicines and those treatment I just mentioned, the burden on our community is very high. And these treatments don't usually end up with people living normal lives. So this atmosphere affects the attitudes of our community towards many topics in health. And of course, these are at a population level. There are lots of individuals with their own views, but in general, people living with rare and genetic conditions have a more permissive attitude towards things that might help progress towards meeting this unmet health need. And that includes sharing data, the use of animals to facilitate research, attitudes towards reproductive choice, and some of the new, so mitochondrial replacement therapy is a reproductive choice technique that has been in the media an awful lot in the UK, and also their attitudes towards innovative medicines. And all of that comes with the caveat that this is in the context of high quality, secure, and appropriate ethically managed research. This attitude comes from the drive that our community has to leave no stone unturned in the search for cures and treatments for rare conditions, and any means to take control of how the conditions affect our community. It's very important to note that genome editing is the topic of our meeting today, but it's certainly not the only avenue that our community wants explored. And they don't see genome editing as a single beacon for the future. There are other opportunities too. So to move on to some of our engagement activities here, the bottom line is pretty simple, that we're strongly in support of exploring the possibilities of genome editing. And here are just a couple of quotes. One participant in our work in 2016 said, "I feel any person with a life-changing or life-limiting condition should be given the chance of a normal life." and that they would be very pleased to participate in research. The views from a year later on what genome editing might achieve reflects the broad potential here, that we can learn more, we can accelerate research, and hopefully make progress. There is some wariness from having been participating in progress in the past, and one of the participants was essentially giving the message that he's been told many times over his life that he's a few years away for a treatment for his condition, thalassemia, and those claims haven't always come good. So our first piece of work was in November 2016, and the community, as I've said, are very broadly supportive here. I'll just pick out a few key points here. That this message came after a series of educational webinars, and one of the messages was that the community wanted to learn more. So there's a drive for information, and I think the research community that know most about this are the best to feed more information to the patient community. There was a topic on human enhancement, and unsurprisingly, the patient community was strongly in favor of focusing on unmet health need. Interestingly, the community wanted to be able to participate in the regulation of this technology and future technologies, and that matches our attitude towards many healthcare decisions. So our second piece of work was even more in depth, with a series of 5 workshops engaging people affected by genetic conditions and people from the fertility sector as well. There were 2 products from this, a cohort of the community who became much more informed about genome editing and went on to speak at various events across Europe on the topic. And that really materially improved patient engagement in genome editing. I believe one of our participants spoke at a very early ARRIGE event, in fact. We also published a report that made recommendations aimed at ensuring the clarity of communication about genome editing, and that report was targeted at those that might in future engage the community. I don't have time to go into all the recommendations we made, but I just wanted to touch on the rationale for taking this approach to the work. That if we can be clear about what we're talking about when we talk about big steps forward in innovation that bring their own ethical debates or quandaries that need discussing with the community they affect, then definition is very important to make sure that we all understand what we're talking about and work towards common ground. In previous work for Genetic Alliance UK, I've seen definition be a major problem around big data and personalized medicine. They've all taken different names at different points in the long-running debates around these topics, and I haven't felt that a clear definition has ever really been available. And I've seen panels where every panelist has a different definition of those topics. Genome editing is a really broad topic with lots of potential uses, lots of potential approaches to delivering the benefits that it can bring. And we really do need to empower the public to have a say by being clear about what we're talking about when we talk about genome editing. And essentially a naive patient view isn't very useful here. The more informed the patient is, the better they can contribute to discussion on this topic. So I'll just wind up by talking through a few things that have cropped up in my broader work that we see coming up in discussions around genome editing. So where there's been ethical discussions around genome editing, I've been surprised to see how much equity of access has been a topic here, and I've seen calls that genome editing only be available if it's available to all. And that's a laudable aim that I absolutely agree with. But it feels like we shouldn't let ourselves be held back by that aim, given the fact that there's inequity across essentially all forms of healthcare access internationally and within nations and between communities and so on. And I've listed a few examples of inequity in access to healthcare provision in the UK, reproductive choice between different conditions, even access to the same medicine between different patient communities is inequitable in the UK and I presume in other areas of the world. We've seen calls for access to be controlled around the seriousness of a condition. And I just flagged the warning here that seriousness is a subjective label, and one man's condition that is easy to handle could, for many reasons, be very challenging for another family. And we need to be very careful about how we draw lines in spectrums and be careful about the consequences of drawing lines in those ways. So seriousness is actually one of the criteria for controlling access to reproductive choice in the UK. And pre-implantation genetic diagnosis is permitted for more than 1,000 genetic conditions in the UK now. All of them have been judged serious in that regulatory process. Now we've seen a lot of ethical concern and debate, and justly so, around genome editing in the germline. And that's right that we have that discussion, absolutely. But we should learn from the fact that a lot of the deliverables that could come from germline genome editing have already been discussed and managed successfully. So most of the reproductive choice opportunities from genome editing are already delivered through pre-implantation genetic diagnosis. And the challenge of introducing genetic material has been covered as well through mitochondrial replacement therapy. And I want to finish just by saying, emphasizing the importance of the patient community in all health debates. And I think most where innovation is happening quickest and is taking greatest steps towards progress, that our community are the ones that benefit most from innovation. And we're also one of the communities that take greatest risks in terms of helping innovation drive forward. So I just finished by emphasizing the importance of engaging the patient community, carers and their families in making sure they're participating in these kind of discussions. And I think, therefore, I'm very grateful to be able to speak here today. So thank you very much.
Thank you very much, Nick. That was a great presentation and very interesting. I believe we meant to keep the questions for after the second presentation, because I've got already a couple for you, but I'm sure others will have as well. Yes. So the next presentation is pre-recorded, as Luis mentioned. So Gaetano Burgio is a geneticist group leader at the John Curtin School of Medical Research at the Australian National University. His current research aims to explore the diversity of the bacterial defense systems against the phage, with an emphasis on CRISPR systems. He also leads a mouse transgenesis facility, which actually generates CRISPR-edited mice. And today, in his pre-recorded presentation, he will present to us the report of the ARRIGE Scientific Committee on Gene Drive. So you have the recorded presentation.
I will be launching the video. It is the volume of the audio is not that high, so you might have to put the volume a bit higher to follow that. Okay.
Okay, got it.
Okay.
Good afternoon, everyone. This is my absolute pleasure today to present the view of the Scientific Committee on Gene Drive. And I would like to thank the organizer to invite me to give this presentation on behalf of the ARRIGE Scientific Committee. Unfortunately, it is rather late in Australia, so I am not able to take the questions in live. However, if you have any questions, please use the chat function, and I will be more than happy to reply to those tomorrow or later in the week. So really an important question that underpins our view is what is gene drives, how important it is, and why as ARRIGE scientific committee we have to come up with position statements, more exactly a policy document regarding gene drive question. And that would be the main part of this presentation. So there are two instances that most of ARRIGE members know about gene drives. The first one is on deadly animals that are mosquitoes. These mosquitoes carry diseases like malaria, Zika, dengue, and kill a lot of people. More than, for example, snake, lions, dogs, or so. The other instance is the emergence, the really widespread of invasive species. And why is it a problem? Is because it endangered native species and affect the entire ecosystem. So it's very, so this is those two instances where gene drive could play an important role, and where all the debate has been held for the last couple of years. And there are some examples there of issue that was brought regarding gene drives. So one is something you've seen quite a lot. If mosquitoes are so deadly, why not to kill all of them? And this is where gene drive comes in. The second instance is, for example, we can use gene drive to eradicate weak pathogen, for example.
So sorry, there must be a stop. So we will be reconnecting. Okay.
Good gene drives. So one of invasive species, and why is it a problem? Is because it endangered native species and affect the entire ecosystem. So it's very, so this is those two instances where gene drive could play an important role, and where all the debate has been held for the last couple of years. And there are some examples there of issue that was brought regarding gene drives. So one is something you've seen quite a lot. If mosquitoes are so deadly, why not to kill all of them? And this is where gene drive comes in. The second instance is, for example, we can use gene drive to eradicate weak pathogen, for example, but for eradicating invasive species. And on the other side, this headline shows that what was bought is gene drive too risky for field trial. So the question is where we are, what is the state of the technology, and what is the ethical issue attached to it, and what are the community engagement to make the technology better understood from everyone. So this is where I believe ARRIGE plays an important role. So before we start really digging into the gene drive question, I would have thought the basic would be to explain briefly what gene drive is. And I have this movie for you showing very well what gene drive is, and the example is on mosquito there. So as you can see here, the classical mosquito gene technology is to replace the DNA. And then it makes the mosquito immune to malaria and breed and spread. Unfortunately, the spread is 50% inheritance, and as long as the generation comes, there are less and less GM mosquitoes. So that means we need to release a lot. So what gene drive is instead of what we do here is to insert the CRISPR cassette there, and the CRISPR cassette auto-maintain and replicate. So what the consequence of this is, it really increase significantly the spread of the gene drive throughout the population. And as you can see here, that kills all the malaria parasites. And it spreads in 3 to 5 generations to the entire population. So this is the really important feature of gene drive technology using CRISPR-Cas technology. So the question is, what is the view of the scientific committee on gene drive? Why is it important? Because this gene drive technology has a strong potential, as just illustrated with the malaria parasite. And there are a series of issues that have been brought on it. So the first, before we go into the policy matters, what I would like to introduce is the process. How we came up with this position on gene drive. So the matter was raised from the ARRIGE boards before the COVID lockdown in March, I believe, 2020. So what happened is it went to the ARRIGE Scientific Committee, so that I'm chairing, and what we came up to basically answer the simple question, what is our view on gene drive? We come up with a working group. So what this working group is part of the ARRIGE Scientific Committee with members that have worked on that question before. So they have deep insight of the gene drive technology. So the working group has a lot of discussion, and we drafted a position statement. And more state of the art really than the position statement. So what happened next? Once we have this draft, we went back to the ARRIGE Scientific Committee for peer review. And it was truly a peer review because it has significantly improved the quality of the document. And then we went for another layer of peer review.
So I'm sorry. It seems that we're having a problem with this video. I deeply apologize. Something went wrong. The video crashed. But then when I tried to reinitiate, the video was not transmitted. So I understand that you did not hear. So since we had the break later on, and because I don't want to sort of kill this presentation by Gaetan, I will restart again because it's only 15 minutes video and I think it deserves to be seen completely. Do you agree, Virginie? All right, so I will start it again. So hopefully this time it will go without problems. Sorry for that. I'll launch it again.
Good afternoon, everyone. This is my absolute pleasure today to present the view of the scientific committee on Genome. And I would like to thank the organizer to invite me to give this presentation on behalf of the ARRIGE scientific committee. Unfortunately, it is rather late in Australia, so I am not able to take the questions in live. However, if you have any questions, please use the chat function, and I will be more than happy to reply to those tomorrow or later in the week. So really an important question that underpins our view is what is gene drives, how important it is, and why as ARRIGE scientific committee, we have to come up with a position statement, more exactly a policy document regarding gene drive question.
So I'm sorry to say that something is going wrong because this is not being transmitted, Virginie, and actually I just don't know why this is happening. So I suggest we can start. This is a bit unfortunate. I apologize for this, but we will be uploading Gaetan's video into the ARRIGE YouTube channel, so everyone will be able to follow this later on. And in the meantime, I think we can have some time to discuss Nick's presentation. So Virginie, thank you, Nick, and apologies again. So just go ahead. So you need to be reactivated. Yes, now you can be reactivated. Thank you.
Great, thank you. Thank you. Yes, indeed, it is unfortunate, but if we could watch the video later on, then that would be fine.
Okay.
Okay, great. Thank you. So I don't know if people have questions for Nick, but I've got a couple. Because it was quite a short presentation, I guess in the sake of time, you didn't really have time to detail the recommendation that you developed following the work that you did with your community. So I wondered if you had to pick a few, maybe in order of priority based on the interest of the audience that we have, which one would it be, and maybe why you think those are particularly important for the audience? You can't unmute.
Nick, we have to reactivate Nick. Yes, now you can reactivate.
Thank you. I felt very trapped there. So I guess there are 8 recommendations from our report. I think I'd start by picking one, which I mean is so amazingly obvious, but didn't actually came as a bit of a surprise to us, that if you're trying to describe genome editing, a lot of your focus to a naive audience needs to be on ensuring they know what a genome is, because the word editing is used quite a lot and people understand what that is. But actually, to understand the potential of genome editing, to focus a significant portion of what you're talking about on what the genome is and why that's important to try to actually achieve the purpose of genome editing. Another important one is on using the term genome editing exclusively. So there are lots of synonyms to genome editing, genetic modification, genome engineering, gene editing, and so on. We pick genome editing over gene editing because you can edit more than one gene, and genome editing would cover one or more. And then just stick to that, because though one might become bored at using the term, it is much easier to understand if you're trying to learn, if people use the same term again and again, and not to use things like CRISPR, because CRISPR might not be the tool that we're using forever. When that becomes obsolete, then we don't want to have to start teaching everyone something new straight, as soon as we start using a different tool, we want to keep the learning that we've had. And so if we stick to genome editing as a name, then that will be helpful. Are you trapped now, Virginie, on mute?
Okay.
Okay, I think I'm good now. Thank you, Nick. Yeah, no, that does make sense. And I also just wanted to maybe insist on the point that you made a couple of times, that you can't approach people cold. And by that, I mean, like you say, the naive patient or citizens, even, and asking them what their views or their opinions are on genome editing. So it's really important to explain, to have the same understanding, as you mentioned, on the terms and the concept and the potential impact, so people can really contribute to the discussion, starting at the same level of understanding. Because if you do approach or consult the patient community or even the public, maybe those will be different perspectives, obviously, but without any kind of explanation, then the latest thing that they read in maybe a not even specialized kind of newspaper might just stuck with them, and that's going to forge their opinion if they don't have the basic kind of understanding of what all the different components to understand what genome editing is and what it could potentially do as well.
Absolutely. I wish I'd said it as eloquently as that, Virginie.
Right.
Virginie, you have a question from Jennifer. Jennifer, you can ask yourself.
You might have to unmute Jennifer, I guess.
No, Jennifer can unmute herself.
Of course.
Okay. Yes, thank you very much, Nick, for your presentation. Do you exchange with disabled groups, patient groups, disabled associations, disability rights groups, who argue against genetic modification because they underline that these practices will stigmatize them or will stigmatize them? I was just curious to know if you exchanged with them.
Yeah, so I mean, we that disability rights argument, and it's especially around reproductive choice, so around germline use of genome editing. Yeah, we have proponents of that argument in the UK. Certainly, it's definitely not a majority viewpoint of our community of 200 patient organizations supporting those living with rare and genetic conditions. And I think the engagement of that community in reproductive choice activity is a brilliant example of how a pro-choice attitude is the best way out of this debate, where we can put the opportunities on the table for people to make individual or couple-based reproductive choice decisions without judging which way people choose to go, and make sure that information is available for people to take that choice, and pick the option that's most appropriate to them. The communities we represent all are focused, or mostly are all focused on the day-to-day care of people living with rare and genetic conditions. So they're spending their lives with people affected by these conditions who are living with them and are helping them deal with the challenges that those conditions pose. So while we have a majority of membership that support reproductive choice, you can see that the availability of reproductive choice and attitude that's positive towards reproductive choice isn't contrary towards believing that people affected by a disability should have a rich and full life and have as much to contribute to society as any other individual in society.
Thank you, Nick. I agree with your response and with the work that Jordis did as well to a smaller scale, because we only had one workshop with patient representative from European federations on specific rare disease or national alliances, so on all type of rare disease. And even within that kind of community, we did, you know, also have a bit of a debate around that. And yeah, opinions and perspectives vary. You cannot have one, you know, I know we say, you know, one voice for the patient community, but on those issues, you have to respect people's preferences, and based on your life, on your story, it's going to change. So that's, again, yes, the pro-choice attitude is the one that we have to go on. But I think for that to still be a possibility, maybe in the future, we do have to educate the public as well, because they are the one that sometimes, you know, have that look on other people and maybe might judge other people's choices. So we do need to educate the public at large, I think, so that that choice can be respected in the future. I have another question, but I think
yes, Virginie.
Yes.
Yes, I can read the question from Milen.
Okay.
The problem of acceptance when you are in need and have innovation therapies as only hope puts a bias on the acceptance of the therapy by the general public. Do you think the equilibrium between change and choice is altered in this situation? It works only in terms of a bottom-up approach, but does not respond to general perspective, or does it? Thank you anyway for your research.
I'm not convinced. Sorry, I'm not sure I follow the question, but I just offer the view that there's a spectrum in views between the public and people with the most serious challenges from disease, whether they be rare or not. And I mean, the spectrum is not two-dimensional. And I mean, I think if you look more into the population, you'll see quite a lot of heterogeneity in the attitudes people have towards innovation in different dimensions. But I'm not sure I'm answering the question.
I don't know if
you on that one, Virginie.
Francois has a question.
Hello, Nick. It's will be a quick question. You have presented various ways to engage patients group, like focus groups or opinion papers that you published. But have you developed also the means to assess the impact of all these activities that you are developing? You have developed, sorry.
I stuck. Yeah. He needs to be unmuted, I think, Louis. Nick would like to respond.
It's okay.
Sorry, it's the learned response in Zoom meetings is to mute when you stop talking. So we have seen our report covered and picked up around the world. Our organization hasn't done as much work on validating the impact of our work as the Progress Educational Trust have in looking at the impact of our findings. I've just got anecdotal responses that I've met people who have thanked us for the report, and that has got to them through organic pathways. When our site went down for a while, an academic at the University of Berkeley got in touch because it was a resource for their teaching program. So we're seeing that it's reached globally, and that's thanks to some learned societies disseminating our report themselves. So we've been pleased with the impact.
All right. I think we have to close and we have to break. So, or maybe did you have a last minute question? There is a last minute.
I did, but he would deserve a bit more time. So it's fine, I think.
Okay, so then, so
Nick
thank you, Virginie, and thank you, Nick. I apologize again for the video of Gaetan. Gaetan's video will be uploaded into the ARRIGE YouTube channel, so you will be able. So we now have a pause. We have a break of 15 minutes, 1-5. In 1-5, we are back again. Thank you.
Thanks, everyone. Thanks, Virginie.
Thank you. Bye-bye.
Bye.
So Francois, whenever you want, maybe Elena, you can, Elena can activate her camera. Okay, you can reactivate your audio, Elena.
Okay.
Okay, ready?
Yes, yeah, Francois.
Okay. Thank you, and welcome to this session. So we all know that the use of genome editing in human is what makes mediatic buzz. However, we should keep in mind that other fields of implementation are also in development, sometimes even in faster manner than the one in human. And that's the reason why we have invited Elena Gomez-Diaz and Ruud Demacht to provide us with updates on two of these fields: the genome editing used in the regulations of animals responsible for vector-borne diseases, and the genome editing used in plant improvement. So Elena Gomez-Diaz is an evolutionary parasitologist of the Spanish National Research Council in Granada, and she dedicates some of her work to the implementation of CRISPR in gene drive systems to end malaria. That would be a good way to complete the few words from Gaeta. And then her presentation will be followed by the presentation of Ruud. Ruud is a project leader of several projects dealing with fruit development and ripening. And also, that's important for us, Ruud is vice chair of European Commission funded Cost Action Planted, genome editing in plants, a technology with transformative potential. So we are very happy to listen, both of you, in 15 minutes for each of you, and questions afterwards. Thanks to Elena. If you can, the floor is yours.
You can start sharing.
Yeah. Okay, so you can see my slide,
Yes.
my slide,
yes.
right?
Yes,
Yeah,
carry on.
Thank you very much to the organizers for inviting me. It's a great pleasure to share with you some insights about the use of gene editing and gene drive systems in mosquitoes, applied to mosquitoes and applied to vector-borne diseases. As you all know, vector-borne diseases are cause of death, are one of the most important cause of death. They are primarily in tropical areas and occur mainly in developing countries, where they are not only a major global issue, but also an are of economic concern. One of these diseases, the most devastating and deadly, is malaria, which despite of several decades of efforts to eradicate the disease, is still causing 500,000 deaths a year, mostly in children. And also it estimated that still affects almost 40% of the total population, so 200 million people a year. So over the last decades, there have been several strategies to end this important disease. Traditional methods to attack the parasite, including drugs and vaccines, but the parasites have evolved all types of resistances. So there are other pack of measures that are aimed to control the vectors that transmit the malaria parasites, so the mosquitoes. The most successful and the ones that have been deployed and contributed greatly to decrease the numbers have been the indoor residual spraying with insecticides, and more recently the use of bed nets that are impregnated with insecticides also. But the problem is that the mosquitoes are also very smart animals, and they have evolved all type of molecular resistance to these insecticides, and also behavioral resistance. So now they're starting to bite during the day and not during the night, when we are protected by these bed nets. So this has led to a huge consensus that there is a huge need for new tools, new measures, and in these next generation tools, are, of course, genome modification of mosquitoes and the development of gene-drive systems, which allow to spread this genomic modification in the population, and that this is sustained over time. There are two types of strategy that have been that are being used or thought in the context of mosquitoes. The first one is population replacement. So we engineer mosquitoes that somehow have a protective gene that avoids or that prevents them to transmit malaria, or that make them less competent for transmission. And the other approach is to introduce in the population engineered mosquitoes that are sterile, either males or females, and at the end, if this gene spreads in the population, will ultimately collapse it. This is one of the most important papers regarding this, the development of these technologies, genome editing by CRISPR-Cas9, coupled with a gene-drive system, is one of the most important, and I will tell you later why. It is of the type of population suppression. So the principle is that targets the gene double sex that has two isoforms and controls sex determination in Anopheles mosquitoes. There are two different splice isoforms, one for the male and one of the female. The one of the female has this exon that is unique to this sex. Then this strategy blocks this exon, and then when this is coupled with a gene-drive system, when the female, this doesn't affect the male, when this female has this modification in homozygosis, then the phenotype is intersex and the female is completely sterile. So these authors show that this modification with a gene-drive system is able to spread over the entire population and reach 100% prevalence in 7 to 11 generation, and progressively reduces the egg production and ultimately leading to the collapse of this mosquito population. This has been proved in a cage mosquito, so in the lab. But the question is that, okay, if we have the technology, if we have a genetic modification that works, and we have a gene-drive system that has been proven to be effective in the lab, can we put this in the context of vector-borne diseases? Can we use this technology to end malaria? And this is the question that this important project is approaching. So the Target Malaria Project is a scientific consortium, an international consortium, non-profit, that is funded by the Bill and Melinda Gates Foundation, and is taking place in 4 different countries in Africa that are major hotspots for malaria transmission. I will focus later on Burkina Faso, that it's where we have been working several years since 2013, and where this project has just started. So this is the video, the promotional video of the Target Malaria Project that is taking place in the Bana district in the south of Burkina Faso. This is how it looks like, and I don't know if you can hear it, but well, they explain.
We see the video, but we don't hear the audio.
Okay, so anyway, they're explaining what this can imply in terms of ending malaria, and which are the first steps that they are taking. So the project is divided into three different phases. The first phase, that is ongoing, that is already taking place, consists in producing engineered mosquitoes that are sterile. So the males are sterile. This is made by introducing a nuclease that will destroy the X chromosome in the sperm and also in the fertilized egg. So all the offspring will be sterile. This has already started in Burkina Faso. They have already released these mosquitoes, and what they want is to collect entomological data by marking and recapture methods. So they will measure how far they travel, how this introduction affects other mosquito species that are present in the region, and which is the dynamics of the Anopheles gambiae population. The second phase that is planned after this field testing is the self-limiting, meaning that they will introduce, in this case, the difference is that the suppression of the X chromosome happens only in the sperm, not in the fertilized egg. So this will cause a bias in a sperm that will be bearing the Y chromosome and not the X chromosome. And at the end, we will have an offspring that is biased by two males. The problem of this is that this modification has fitness costs for males, because there will be a lot of males, fewer females, and then there will be a lot of males that will not reproduce. So at the end, the mosquito population will be suppressed, this modification. But so this is what they plan the next. And the third phase is the self-sustaining, meaning that they will couple either this strategy or the strategy that I explained you before with a double sex gene that causes infertility in females with a gene drive system. So this modification will be spread in several generations, and at the end we will have a collapse of the mosquito population. So this is what they plan in the long term. This is another video that I will not put because you cannot hear it, but basically talks about the promises, and this is something that is reflected in the report, the ARRIGE report. Let's say, no matter which are the risks, but the benefits are huge, because we are talking about one of the major diseases of the humanity that has not been solved yet. So if you speak with anybody in this region and told them that with this technology we may eradicate malaria, they will say yes, no matter the cost, let's do it, right? So in principle, the benefits are enormous and overcome the risk or the limitations that we may thought about. But the realities as scientists, we have to think about those risks, because there are many, and there are many because this technology is relatively new, and the reality is that gene drive systems, and in the context of mosquitoes, we have very little experience of how they will respond and evolve in the wild. There are several limitations. I will briefly focus on three that I think that are relevant in the context of the Burkina Faso, where this project is taking place. The first one is about ecological complexity. Malaria parasites are transmitted by more than 30 different species of Anopheles. Only in this Bana province in Burkina Faso, there are 3 different species of Anopheles present. This target malaria project targets only one of them. So what happens if we suppress one of the mosquito species is that the parasite will switch preference to the other 2. Okay, so that's a possibility that has to be taken into consideration. The second, this is what is illustrated in this slide, the different mosquito species present. The other problem is that is about confinement, about how we can control these gene drive systems, because in theory it is a local deployment of gene drives that will have a local effect. because in principle, mosquitoes were thought to travel short distances. But according to recent studies, for example, this study that was published last year in Nature, millions of mosquitoes travel hundreds of kilometers by the prevailing winds, transmitting malaria. So if we introduce those genetically modified mosquitoes, they may also be able to spread and to travel those large distances. And this is something that we have to take into consideration. And the third limitation that I would like to comment is about the problem of gene drive is not only a scientific problem or a scientific issue, is also a social and political issue. And this is something that in the case of the Target Malaria Project in Burkina Faso is causing a lot of problems, because there are several associations, the civil society that claim that there has been no consent from the civil society for the start of this project and for the release of these mosquitoes, and that there is no acceptance of this new technology in the region. So there is a lot of fear, there is a lot of misconceptions, the knowledge is very limited. So there is enormous work in order to approach this problem from an interdisciplinary point of view. Some people talk about the post-normal science, a science that is not only about the limitations or the challenges of the technology, but all the social and the political challenges that it implies. Politics, journalists, and so on. So this is, I think that is a good end for keep you thinking about questions and to start the debate after the next speaker. So thank you very much for the time and for the opportunity to talk a bit about mosquitoes and malaria. Thank you.
So thank you very much. I'm sorry, my microphone was muted. Thank you very much, Elena. So as we proposed at the very beginning of this meeting, it's the question will be collected and asked at the end of the session. So let's move to Ruud presentation, please. Ruud, the floor is yours.
I'm not going to start my video yet. Okay, there we go.
Okay.
Okay, thank you, Francois, for the introduction and for the invitation to speak today. I'm not going to talk, at least not much, about my own work, because today I'm representing the cost action plan there. So, but I prepared just a little introduction about myself. I'm working in Wageningen plant research. So it is confusing, I realize, but there are two entities, Wageningen University and Wageningen Research, which is the former agricultural research. So,
Ruud, this is you. You have to restart sharing the screen because something is wrong with the screen.
Are you seeing anything?
Yes, we see part of your screen, but it's kind of frozen.
Yeah, I have to try to get to resume sharing. Just a moment.
Maybe you stop sharing and you try it again.
You seeing slide 3 now?
Well, we still see your presentation. So just go to the first one and launch it.
Okay. I'm sorry about that.
No problem.
I practiced it, but I can't see what you are seeing, obviously.
So you can share your screen now.
I'm going out of the presentation.
All right, then you launch the presentation.
Yeah, I'm afraid that if I make it full screen, it won't work, but I'm going to try once more. The problem is that
what happens is that you're probably using a Mac, and
yes.
and you're probably using the presentation mode, and this is why. So in this case, I suggest you just you advance the slides as they are shown here.
Yeah. Can you see it now?
Yes, we can see. Thank you.
Okay, yeah, I'm sorry about that.
No problem.
We noticed that Mac has some problems with programs that other people don't have. Okay, so I'm working in Wageningen plant research, which is part of Wageningen University and Research. And as Francois already mentioned, I'm studying, among other things, plant development, specifically tomato fruit development in ripening. And we started using CRISPR-Cas mutagenesis in tomato about 7 years ago. But first, about gene editing or genome editing in crops. So editing in crops started way before the invention of CRISPR-Cas mutagenesis. In the mid-90s, other nucleases were used, like zinc finger nucleases. We have, from 1999, the oligonucleotide-directed mutagenesis, which one American company is using and commercializing. In 2009, tail nucleases were introduced, and the company Calixed is commercializing products from that. So actually, the two products that are now on the market that are gene edited are using these technologies. In 2012, as you well know, CRISPR-Cas was discovered, and in 2013, this was first applied in plants. So probably like in other organisms, we see three main applications of gene editing in plant breeding, which are called SDN1, 2, and 3 for site-directed nuclease, to include also the older nucleases. And SDN1 is causing small deletions or insertions that interrupt the function of genes, with which you can delete unwanted traits in plants. And we could see this as replacing chemical or radiation mutagenesis that has been used already for tens of years. SDN2 is the substitution of one gene version, an allele, for another one by making a limited number of changes. So that could, for example, include base editing or prime editing. And it resembles the backcrossing for integration of traits from one cultivar or one variety into an elite variety, which takes a couple of generations to achieve. And with gene editing, this is much faster. SDN3 finally is the insertion or replacement of entire genes, including replacing with genes from other species. So then it starts to resemble transgenesis somewhat, or from sexually compatible species. So advantages for breeding in crops is speed, precision, and relatively low cost. But I should make a disclaimer that many things that are claimed that we will be able to do very soon are quite challenging for CRISPR-Cas. For example, I've been working with insect-resistant crops that contain a gene from a bacterium, Bacillus thuringiensis, and it will be difficult to achieve something like that with simple SDN1 or SDN2. So what could gene editing in crops be used for, or is it already being used for? That is, for example, the removal of gluten allergens in cereals like wheat. That's been done in our own institute, but also in Granada in Spain. Improving seed oil quality and make it healthier or better to cook. And Calix, the soybean oil, is commercialized right now in the US. Then there is not the best example, I think, but there is herbicide resistance, and Cebus, the company in the US, has produced an herbicide-resistant canola and is commercializing it right now. Maybe a better application is deleting susceptibility genes that a pathogen needs to be able to infect or invade a plant, and if you knock them out or change them, then the plants become resistant to pathogen infection. And the USDA has just approved a blight-resistant rice variety that was produced with CRISPR-Cas. You could improve the agronomy of existing crops, like flowering time, the stature of the plant, or the yield. Reduce food waste, for example, anti-browning in mushrooms or apple. Increase the shelf life so that you don't have to throw away produce. Mitigate some of the effects of climate change, like heat, salt, drought stress tolerance, or de novo domestication of wild plants or crop relatives, like producing new crops that already contain stress resistance instead of trying to change existing crops to make them more resistant. This is one of my favorite applications, and one example from my own field, not my work, is the domestication of tomato. Cultivated tomato was domesticated over several thousand years from a wild relative, Solanum pimpernellifolium, shown on the left, to produce cultivated tomato, Solanum lycopersicum, which is not so stress tolerant, but it is optimized obviously for horticulture. Like in some other crops, we know fairly well the major genes that were involved in domestication and that give modern tomato their properties. So researchers did CRISPR-Cas mutagenesis in Solanum pimpernellifolium for 5 to 6 genes in several groups, and in that way produced a de novo domesticated tomato, demonstrating that CRISPR-Cas mutagenesis allows you to do something like this in only a few generations. So the development of crop gene editing poses a problem for the perception by the public, the regulation by the EU, and the enforcement of those regulations. So current EU regulations stem from 2001 for the release into the environment, and they did not foresee such fast new developments. Also, our understanding of what is natural and of the plasticity of plant genomes has grown tremendously as more genomes are being sequenced. And we realize that also in nature, a lot of things change in sometimes very bizarre ways. Many of the edits in crops will also be indistinguishable from natural variation or from changes made through exempted mutagenesis methods like radiation and chemicals. So a lot of my colleagues and myself expected that the European Commission would treat these edited crops with small edits like the exempted mutagenesis methods. Unfortunately, the European Court of Justice decided differently. So it ruled that products from targeted mutagenesis like CRISPR-Cas or ODM are still regulated under Directive 2001 for the deliberate release of GMOs, while products that are derived from undirected mutagenesis, which cause many more mutations, unknown mutations, much larger effects, they are considered GMO, but they are exempt from regulation because they have been safely used for already years. And CRISPR-Cas doesn't have that claim. Enforcement will be challenging because of small changes that are difficult to detect. The Commission has no intention to change the directive, although many scientists argue for that. And the European Council of Countries requested a study from the Commission about the status that is due early 2021. So we're very anxious to see what they will tell us. In the rest of the world, regulation is either process-based, as in the EU. So if you do genetic modification, it falls under the regulations, or product-based, where actual product is being studied and its properties. So new developments in gene editing in crops, some have already been mentioned. New Cas nucleases like Cas12a are already being used in plants. Base editors are being used and they work efficiently in plants. Prime editing is just starting. So replacing small stretch of nucleotides is just proof of concept. And template-guided repair, homology-directed repair, is still very challenging in plants. So now switch to the cost action plan that. The cost action plan that is being financed by the Cost Foundation, and it is all about plant genome editing and assessing the full innovation potential and impact as one of the objectives, set the direction of future research, to promote the link between research and innovation in a socially responsible manner, and to examine the synergistic interactions with closely related fields like genome sequencing. Some numbers. The chairperson and the grant holder is Swedish University of Agricultural Sciences, and the chairperson is Dennis Eriksson, who asked me to give this presentation. We started last year. We already have a lot of participants from all European countries, but also neighboring countries and further afield internationally in the rest of the world. And it includes various stakeholder groups. The action has 5 working groups. Working group 1, technical platforms, is the one where I am the vice chair. There's also impact, policies and regulations, perceptions, and an overall spanning communication working group. We had our first meeting in Serbia, Novi Sad, in November last year. We were supposed to have another one in September of this year, but for obvious reasons that didn't go through. Hopefully next year we will have the second meeting. The action is involved in some of the consultations that are being held by the European Union and EFSA. And it organizes training schools. There was one about regulatory options in January 2020. There was supposed to be another one in March, April of this year about genome sequencing. But for the same reasons that didn't go through. So hopefully next year. A cost action finances collaboration, but not direct research. However, it does finance short scientific missions where a member researcher can go to another lab in the consortium in another country and stay there for up to 3 months to do research or to learn things. And the action is active on a number of social platforms like YouTube and Twitter. We have a website, and we welcome proposals for collaborations and joint events. Thank you.
So thank you very much. So we can start the question.
You can stop sharing now, Ruth, and Elena can reactivate the video.
Okay, so there is a question for Elena from Veronica Popa. How can you be sure that the impact of this strategy will only be local? And how will you mitigate the impact on the trophic chain?
Yes, actually, this is one of the questions that I think that this doesn't have answer yet, because as I tried to illustrate in the talk, there are evidences that mosquitoes can travel and can go, and there are no frontiers for them to go from one country to the other. And also in terms of the fact that there are more than 30 species of mosquitoes that transmit malaria, then the solution is, okay, should we kill them all? because as the question puts the emphasis, they also play an important role in the ecosystem and the ecosystem chain. So this is not the solution either, to kill all the mosquitoes of the world, of all the species. So this is something that we will have to be addressed. One of the recommendations, the current recommendations, is first to deploy regional regulations, regional and international regulation for this type of gene drive systems that apply to mosquitoes. And because this is not that local. And the second is that always a gene drive has to have a B option, like either a reversal drive, so you can stop the process anytime that you need, or a system that is like self-exhausting. So after a number of generations, it will disappear by itself, like the daisy chain option that has been published recently and so on. But this is not being investigated yet in the case of mosquitoes.
Okay. So I don't see another question on the YouTube chat. So I have a question for you. So since the very beginning of this meeting, we all agreed that public engagement is the only way to make genome editing to develop well and to be accepted by the population. So, as you mentioned in your presentation, there was a lot of discussion in Burkina Faso. There are a lot of ONGs like Terre en Vie who are arguing that the consent was not provided properly, as you mentioned clearly. But I wonder how much of the Target Malaria project is dedicated to educational program or to community engagement, because there are a lot of money spent for scientific development, but how about social development?
Yes, this is a very good question. If you go into the page of Target Malaria, they put a lot of emphasis on the efforts that they are making to communicate to the general public and to have this consent and this acceptance. But the reality is a bit different. So in the field and in the places, there is a lot of opinion again. So I think that the problem is the following, that this project is led by foreign institutions and is being implemented by local researchers, right? So up to which point the information is being appropriately delivered, or there is a good program in place taking place, I'm not sure about it. So it's among the objectives, but I'm not sure that it's being implemented in the dimension that it needs to be. Like, okay, because it's this Bana district, but and the project says that they are educating people about mosquitoes and about engineered mosquitoes, but this is very local. It's not a big, an ambitious project to a real and effective engagement strategy. This is my opinion.
Okay, if I can follow on that, we've learned from Ebola crisis that the presence of anthropologists or social science experts is crucial in the way that to facilitate the implementation of new therapy or new behavior. How about Target Malaria? Do you have social science project funded through Target Malaria?
As far as I know, no. So this community engagement is led by the local people that are implementing the project there, but the same scientists, but that are explaining the research and are giving talks and seminars and so on. But there is no like a specialist, like journalist or a a campaign by radio or television or involving social organizations that help to transmit the message.
Hi, Elena. This is Luis. Perhaps the thing is whether you have to train the trainer. So whether you are training these locals in the way that they could deliver the corresponding messages to the local population.
I mean, really, you should take look at the lesson learned from this Ebola crisis, definitively.
Yeah, I think that it's something that scientists don't have. We don't have the capacity to arrive to that level of communication. I like to understand the social, the politic dimensions, the tempo, when and how long, and which are the target. For example, in Burkina, villages are organized in the way that there is a chief of the village, and all the people follow that person. So it is very important to understand how the communities are organized and how they function in order to deliver adequately the message. If the project doesn't have the acceptance of this chief of the village, the whole locality, the whole population will be against, for example.
Okay, thank you. There is a question for Ruud, always from Veronica Popa. How is an unwanted trait defined, and how is the impact of its modification being measured on a long-term ecological level?
Yeah, sorry, unwanted
trait.
Unwanted trait. An unwanted trait is one that is well, how to say that, is amenable to improvement for agriculture. So just to give an example, wild grasses from which cereals were domesticated, they very easily lose their seeds because that is profitable for their spread, but it's not profitable for the farmer. So farmers during domestication have selected variants that do no longer fall from the plant and can be harvested and then be trashed later. So that is, in terms of agriculture, is an unwanted trait. The loss of seeds is an unwanted trait. This is, of course, strictly from an agricultural point of view, not from a biological point of view. And also you have to remember that agriculture is not really ecology. Considerations for agricultural plants are not the same as for nature preservation or for ecology. So I know this is a very big discussion, and a lot of groups are active against GMOs, and recently GMOs are being called gene editing is being called GMO 2.0. So the same kind of discussions will probably come. But you have to remember that there is already a lot of variation in nature and also in agricultural varieties and cultivars that we hardly know and which are much more invasive, much bigger than the small changes that you make with gene editing.
Thank you. Jennifer, do you have a question? Can you? Someone has to activate her microphone, please.
Thanks.
Yeah.
I have a question for Ruud. It's probably naive. I'm quite sure it is. But have there been studies on consumers of these products, these tomatoes, for example, who have consumed them? Have there been studies on their organisms?
Have there been
on people who eat these tomatoes?
In fact, there are a lot of different opinions in Europe and around the world. So
that I understand, yes.
Food products made with gene editing, they are not, well, except for the soybean oil, not much is on the market right now. But if you, for example, in Europe, interview people, as you probably know, it depends a lot on the kind of questions that you ask. If you ask, do you want genetically modified food when DNA has been changed, the large majority says no. But if you ask them, do you want a food, a tomato or something else that tastes better, is better for your health, protects against heart problems, is better for the environment, and so on, then you get a different answer. So it's still very much under debate. It's genetically modified, or in the future maybe gene-edited foods are unpopular in some parts of Europe, but that differs. In the US, that is much less an issue with the majority of people, I believe.
Yes. Thank you.
Well, thank you very much for both of you, and we'll go now to the next session. Thanks.
So Alta Charo and Eric Mesling, you can reactivate your cameras, and we will be reactivating your audios, and also Hervé. Yes. Now you can reactivate.
Okay, thank you very much. So we are going some way to continue on this topic of how to communicate and how to govern genome editing, with some focus this time on human genome editing. We will have the pleasure to welcome first Eric Mélin and then Alta Charo. So I'm going to say a few words first of Alta Charo. Alta is the Warren Knowles Professor of Law and Bioethics at the University of Wisconsin. She's a member of the US National Academies and co-chaired the Committee on Stem Cell Research and Genome Editing. And with Jennifer Merchant, they were involved in the first report of the Academies on Science and Society pillar. She was also a member of the Presidential Commission of Bioethics, and now she serves on the WHO Advisory Committee on Global Governance of Genome Editing, and several committees, so much I can't tell you. Eric Mélin was trained as a philosopher and a bioethicist. He is currently the president and CEO of the Council of Canadian Academies that gathers 3 academies. He was the founding director of the Indiana University Center for Bioethics and associate dean for bioethics at the Indiana University School of Medicine. He's currently a professor at the Danalana School of Public Health, University of Toronto. He was involved in many committees, here again at the Presidential Bioethic Committee of the US. And it's an honor for us to let the floor now to Dr. Mélin for 15 minutes and then Alta Charo for 15 minutes. Thank you for joining us.
Thank you very much. It's a pleasure to be here. I wish we were all able to meet in person, but as we know, the world is not allowing us to do so. So I hope everyone is well in their own homes and with their friends and family. It's a pleasure to be with you, and I'm going to just speak for a few minutes, not to use slides, partly because I think it's one effort to try and humanize our discussion. I'm not going to be presenting data. The title that I gave myself was Reflections on Emerging Developments, which is about as abstract and non-convincing a title as you might wish to think. And that's largely because I want to reflect on a couple of things that have been moving relatively quickly in this field. I really just want to make 4 points, probably obvious to everyone, that this topic, not just CRISPR-Cas9 technology, but genome editing and the entire genomics field has been moving at a rather breakneck pace for probably the last 20 or 30 years, using the very unscientific method of going on to Google. If you just type in gene editing today, you'll get about 239 million hits. If you type in genome editing guidelines, you'll get another 100 million hits, and on and on. There is no curation to this flow of data or who gets to claim what. There is no quality assurance. There's just lots and lots of information in an open environment, which makes it quite difficult to make sense of what is useful, what is evidence-based, what is speculation, what is anecdote. Now, this is not just an observation about the risk of misinformation, but rather it is explaining to us a little bit about the specialization process that is occurring in this field, and indeed a subspecialization process. There are experts now in the very granular areas: plant, animal, human, governance, clinical trials, quality assurance, health technology assessment, and governance. This is no longer a time when one person can be competent in or knowledgeable across fields. And in fact, governments and civil society are facing this problem of how to interpret this massive tsunami of information, whether it be on ethical, legal, or social issues, or whether it is on the data itself. This, on the one hand, is worrisome, but I have a bit of an optimistic gene, and I see it as somewhat democratizing, that there is more information available. It's incumbent upon us, of course, and organizations like this to be very clear about what information does pass the test of, if not peer review, then at least high quality review. And I say this just as an opening first remark, because today we're having this conversation, and just a month ago, it probably would have been a slightly different conversation. Maybe 2 months ago, it would have been a different conversation. Before, for example, the Nobel Prizes were handed out, we might have been having a different interaction about the role that science is going to play, or the publicity of this topic. The second point is that there are many organizations weighing in. My dear friend and colleague, Professor Charo, may be speaking to this, so I don't want to step on her toes. But she, as you announced in your introduction, and others on this in this organization are involved in many committees, panels, working groups, and other organizations. I didn't count the number. One of our colleagues in the field, Francoise Baylis, has written a paper that describes what the laws are in many countries. So this is a very active area of work with lots of organizations weighing in. This itself is gratifying, but like the data point I just made a moment ago, there's a kind of incommensurability between the kinds of organizations that are claiming knowledge of or expertise in, and those who are just weighing in because it's interesting or curiosity driven. So of course there are governments and many orders of government, not just federal or national governments, but I live and work and have lived and worked in both Canada and the United States. And there are quite a number of differences between how provincial governments in Canada and state governments in the US think about, consider regulating or legislating any of the aspects of genomics, let alone the particular aspects of gene editing. There are regional groups. Of course, the European Group on Ethics is well known to this group. ARRIGE itself would be considered an organization that has high competency in this very area. And of course, we have professional science organizations. You mentioned the one that I work with, the Council of Canadian Academies, a bit of a mouthful. We like to think of it as Canada's little cousin to the US National Academies. It's much smaller, but it does do the same sort of thing. It's an organization of the learned societies, or three of the learned societies of this country: the Royal Society of Canada, the Canadian Academy of Health Sciences, and the Canadian Academy of Engineering, not dissimilar to what the US National Academies has with their triumvirate. So there's lots of different types of groups weighing in, and that's not to leave out advocacy organizations, patient care organizations, NGOs, public-private partnerships, the private sector. It's quite a busy environment. I mention this because there seem to be more and more participants in the domestic and international conversation about these technologies. What I do want to make an observation about, and I think it came up in the Gene Drive session just a moment ago, is the need for us to ensure we recognize the global north, global south, to borrow a kind of arbitrary distinction, discussion. There's a risk, of course, that focusing only on the topics of this area of science as a matter for technology development and healthcare access, or even agricultural or animal welfare issues, are limited to the global north. This will be an enduring bioethics and I think social challenge that we're only just beginning to appreciate. I said I had 4 comments. I'm going to raise my 3rd one now. And it sort of flows from the long list of organizations. And of course, organizations do a number of things. They may write guidance or standard operating procedures, professional practice standards. The list of instruments, in other words, of policy development, is probably as long as the number of organizations who write them. Now, of course, no 2 documents have the same force. Some of them may have legal force, some may have moral force, some may have public relations force, and some may have commercial or other private power. What it suggests, though, is that in the absence of a kind of unifying theme for how to organize, make sense of, and harmonize the different documents, what we are left with is quite a significant volume of statements, declarations. There will soon likely be treaties, I can imagine, that are emerging. Just the 2 organizations that you mentioned in Altacharo's bio sketch, a WHO committee, a National Academies Committee, 2 very distinguished organizations. And while many know their counterparts on the other committee or were themselves on one of the other committees, we don't have a World Science Court. We don't have a World Ethics Court. We don't have a kind of meta governance structure that can adjudicate differences between them, legitimate differences, but adjudicate differences between them. This is not a problem that must be solved, but we are in this almost ironic position of kind of a candy store in governance. You know, many years ago, we were looking for guidance documents, and there were very few. You know, back in the days of the early recombinant DNA technology debates, when now everyone is quite aware of the Asilomar meetings in the middle 1970s, producing a document that was at the time one of the few in the world. And now our problem is not looking for documents, it's that we have hundreds of them. They're coming out almost daily. I'm exaggerating because I know it's late where many of you are, and I want to make sure that people are paying attention. But we do have many documents without an adjudicatory mechanism for distinguishing between them. This is really important for a couple of reasons. And one of them flows from the recent human heritable gene editing statement that came out just a little while ago. My good friend Kevin Davies, the editor of the CRISPR Journal, put together a compendium of comments from many people around the world. I happen to be one of them. We were given 300 words. What do you think about this report? Well, for those of us, particularly people like me, for whom 300 words is a throat clearing exercise before I get to say anything, there's not a lot that you can say in 300 words about a report that is not only thick, but comprehensive. And yet the careful reading of that National Academy Royal Society report is, I think, required reading because of the nuance, because of the kind of pragmatic shift that you start to see in ethics and governance towards what would be the conditions under which we might permit this, rather than never shall this occur, it should never happen, the end result of the deliberations were no. I think it's important that the documents themselves, not just because of their volume, requires the careful attention of both civil society and the public, but requires a reading in chronological order. Many of us are aware of and may even have been involved in debates and discussions on a lot of these topics. But it would be irresponsible to simply pull one out from 10 years ago and sort of use that as the standard or received view. I find it is quite difficult to keep up with the latest position, especially when it is a nuanced position. We will wait with great interest for my friends at the WHO to come out with their statement. I don't know that they will be speaking directly to the National Academies Royal Society statement. It would be odd if they didn't note that it existed. We're all reading everybody else's work. So I think this creates what I would call a challenging environment. Let me close with my fourth and final point. And this is sort of going back to my roots in philosophy and bioethics. And this presentation is actually going from probably the most evidence-based to the most philosophical. So I started with there's lots of data out there. Let me just finish with a similar comment about there are lots of ethical perspectives out there. And those perspectives are often and have historically been captured with endless lists of ethical principles. There's a long history in the field of bioethics where the listing of principles is seen as a sensible and pragmatic strategy for explaining difficult moral problems. There have been pushbacks on whether that approach to even using principles is sensible or not. That's not my point. In fact, the list of those documents that I mentioned oftentimes contain sets of what is now becoming quite a comprehensive list of principles. I'm not saying that people are picking and choosing the ones that they want, or that there is a limited set of ethical principles from which various groups decide which they want to adopt for their own particular use. And I'm just going to read from my own notes here, a comprehensive but not exhaustive list of the principles that have been described just in the last 2 or 3 years in the last few international documents include the principle of precaution, consent, autonomy, balancing risk and benefit, safety, harm reduction, social contract, justice, access, fairness, shared benefits, avoiding discrimination, protecting the vulnerable, choice, reciprocity, and transparency. I have left out many others. So it's not enough to simply say the way that we ought to move forward is with good evidence, which of course we must have, with a good moral foundation, no one disputes that, and with good public engagement and awareness, because this can't be limited to the experts. So I would close, and I'm surprised that even I can get this done in exactly 15 minutes with 51 seconds to go. My perspective or reflection on emerging developments is that this doesn't end. Today's meeting is just the first story or the first chapter in what will be a conversation that's going to happen tomorrow. And when the WHO report comes out, this organization will have another discussion and many others will, and it will keep going. I rather like that. I will finish on the optimistic note that I think this is exactly what should be happening with a fast-moving technology. We will never just answer the question and say we're done and move on. We're now at the point where we've got using these various instruments of policy and the internet to get more information. Thank you very much.
Wonderful, wonderful timing, Eric, and very thoughtful presentation. So much to discuss. We let the floor immediately to Alta, and we will have 10 minutes of question at the end of your two talks.
Thanks very much. Let me just pull up some slides here for you. First, that was a wonderful segue. Thank you, Eric, and thank you, Hervé. It feels like meeting with old friends from old committees here. I wanted to speak directly to what's currently going on and what may happen next, specifically on governance questions. So as Eric was just saying, it's important to begin with a recognition that there really is no mechanism for unified policy or global governance in this area. There are some exceptions in the form of regional agreements, such as the Oviedo Convention, which was developed under the auspices of the Council of Europe, as well as treaties or other formal instruments. But these really are representing only a minority of the countries around the world. And in addition, even once these exist, they still need to be translated into domestic policy through domestic law in most cases. And as we know, countries can have reservations or other kinds of exceptions made to their compliance with these. And so it would not be realistic to rely entirely on these mechanisms to try and achieve some kind of global rationality with regard to this field, because I don't think that uniformity is either possible or necessarily the right goal. If you want to get a sense of what's going on around the world, the Genetic Literacy Project has a wonderful website where you can actually click on any country and take a look at what its current rules might be, and it is kept fairly up to date. So in addition to Professor Bayless's recent article documenting what is currently known about law in these various countries, this allows you to do a kind of ongoing update. The other thing that's interesting here is that you get a chance to look not only at human and health, which would cover things like stem cell and germline gene editing questions, but you can also compare it to the attitudes around gene drives or agriculture in the form of genetically engineered foods and genetically engineered animals. And it's useful to do that sometimes in order to try to better appreciate what the underlying driving forces are, politically speaking, in any one country. In some countries, it seems like it's really about anything having to do with genetics in particular, often having to do with a very particularized history. Germany is an example of a country that is still working through the trauma of the mid-20th century experience with the Nazis, making it particularly cautious about genetic technologies. And then you get other countries where really, such as in the United States, it's more about a religious conflict and about changing demography in terms of religiosity and secularism, in which case the focus will be more on the human health applications, but much less controversy around some of the environmental applications. And this can be helpful in trying to predict where the policy will go. Understanding what the underlying forces are in any particular country is a good place to start. But let's take a close look at least one example of global governance, which is the Oviedo Convention, which really aimed to say that you basically cannot do anything that's going to intentionally introduce a modification that will be heritable. This is something that is very specific and fairly narrow in its focus, but it has had some real influence. So one of the results is that in countries that are partners to this, you have a highly regulated situation, and with germline in particular, completely illegal. The UK's exit from the European Union will open up new questions about whether the UK is going to go in a different direction from the rest of the continent, politically as well as legally. In China, what we discovered is that we have very unclear regulation. There are a series of laws in China that have to do with human reproduction and stem cells that are of overlapping coverage, but don't create a comprehensive framework for germline editing or for any kind of editing. We also learned that although there is oversight of human subjects research in China, it's very disorganized. You have literally thousands of individual research oversight committees at institutions and in regional provinces, but there's very weak control at the national level. And there is a wide variation in the understanding of the applicable rules, if there are any applicable rules, as among these different research oversight committees. And the result is that you have widely varying notions about what is acceptable from place to place in China, as well as many places where research can fall through the cracks and not be completely overseen by any one committee at all. The US has a system in which we have a very highly regulated area for somatic editing under the usual rules for cell and gene therapy. They go through pre-market clinical trials. They have to be approved by a federal agency, and only then can they be marketed. And then there are restrictions on how one can market them based on what they are proven to be safe and effective for. In the area of germline regulation, although Françoise has characterized the US as saying it's illegal, I think that's a little bit misleading. Because of some of the limitations on federal power in the US, it is quite common that instead of saying something is illegal or illegal, is legal or illegal, the federal government will simply say, if you're going to take federal money, then you can or cannot do certain things. And so through the power of the purse, as we say, the power of money, you can place conditions on what people can do. And where federal money is really the primary source of research funding, that can effectively occupy the field, but it is not technically making something illegal. In our case here, through reasons of technicality, what's happened is that we have a budget provision that prevents the federal agency from reviewing and giving approval to any kind of clinical testing of germline editing. And so for the moment, you cannot do it here, but the moment that budget restriction is removed, it now becomes again a situation in which germline editing is legal, speaking from the federal point of view, and is subject to state laws for more regional regulation. And we're all familiar with what happened in November of 2018 when we had this announcement out of China of the use of germline editing on several embryos, and that there had been the birth of 2 children who had been conceived with edited embryos. As it turned out, neither of them got exactly the edit that was intended, which is precisely one of the things people feared, because we could not be sure exactly what condition had been created, and we've not had any real public follow-up of this. China has announced that it plans to tighten up its regulations, and its investigation of this particular situation did reveal that it was done outside the bounds of permissible law. But it's worth noting that the law was so unclear that in the end, what Dr. Hu Jiankui wound up being convicted of was not violation of a research law or a reproductive technologies law. He was convicted of practicing medicine without a license, which says something about the confusion and lack of comprehensiveness of the law in China. So where has this led us? It has led us now to a situation in which we have many different organizations, as Eric was saying, and most recently, the International Commission on the Clinical Use of Human Germline Genome Editing, which was organized by the US National Academies and the Royal Society, but had input from academies of science and medicine in France and Germany and Japan and other countries. It focused on a very narrow question that came out of the Hong Kong meeting. It was this: we are not going to assume that this is going to be legal everywhere. We assume, in fact, it will be legal and illegal in different countries at different times. But if there's any country on the planet where this is going to be legal, then the next question is, how do you make it responsible? How do you do it in a way that meets ethical and scientific norms and can be done without imposing great harm on people in the future? I don't want to spend too much time on them, but I do want to note that their focus was primarily scientific, looking at specifically what kind of research you need to have in the laboratory models, in the animal models, in order to be able to predict the effectiveness and the safety of any particular edit, the stability of an edit over time, and the risks of unintended effects. So they were focusing primarily on science, but they also had a bit of a walk into the terrain of governance as they tried to explain what kind of oversight would be needed to make sure that this science is being done. So their recommendations at the end many of them not terribly surprising. For one thing, that we are nowhere close to being at a point where this germline editing could ever be done properly and responsibly, even if it were legal someplace. And so they actually replicated some of the recommendations coming out of earlier commissions about the need to limit this in the future, if it's ever done, to areas where the need is compelling so that it can possibly justify what residual risk exists after the scientific research has been done. They made a number of comments about the capacities of a regulatory body to actually review work and then follow it up over time. And then most interestingly, because Eric said we don't have an international science court, they did talk about something that would be functionally an advisory group on a global scale to advise on the state of the science with regard to germline editing and feed that information on an ongoing basis to any policymakers deciding whether this should be legal, and if so, how it should be regulated. That committee does in fact feed into the World Health Organization committee that Hervé and I are currently serving on. Its focus is broader. It's looking at somatic as well as germline editing. It started with this notion that we should be in a position to know what's going on. You cannot do good policy without good facts. And so the first step was to start taking advantage of international clinical trial registries to find out, at least in the area of somatic editing, what's going on because of the need to be able to track some of its more controversial uses, including potential use for so-called enhancement, which, subject to another discussion, is very difficult to define. Ultimately, we want this registry to also collect information about germline editing so that we're not caught by surprise by people like He Jiankui or by the people in Russia who have announced that they're interested in doing this, or in Saudi Arabia. But more importantly, I think that the WHO committee is looking not only across borders at the possibility that different countries will have different policies that must be coordinated, and not only that it's looking at somatic therapy for enhancement. I'll talk very briefly at the end about in utero and epigenetic editing, but it is noting that there are special problems precisely because this is a transnational issue. How do we anticipate the movement of patients and scientists across borders? And how do we avoid recreating what happened in stem cell research, where some countries were so poorly regulated that a number of clinics could advertise services that frankly were never proven to be safe or effective and draw patients from all over the world, so-called stem cell tourism. Here we worried about it becoming gene editing tourism. At the same time, countries were sometimes regulating what their scientists could do, and yet they needed to move across borders to have freedom to collaborate. We also are looking beyond formal regulation, which is only one part of governance. We're also looking at the broader ecosystem that includes journal editors that insist upon evidence of ethical review before publishing materials. We're looking at the role of insurers who evaluate risk and will often stop activities from occurring because they simply refuse to insure them against liability for having caused injury. We're looking at the role of professional societies, not only to educate and advise their members, but potentially to be a place where people who have concerns about work in another laboratory, like He Jiankui's, have a place to go and report their concerns and provide a venue to discuss those concerns. When we look into the future, I think it's important to recognize that somatic editing is really where the action is going to be at. Germline editing, even if proven to be safe and effective, will be used for very few things. But somatic editing has many more interesting possibilities. Enhancement to make somebody pain resistant if they have terminal cancer is wonderful, but it also could be used to make military people more resistant to pain, which might be a little bit concerning. We can look at somatic editing of the fetus in the uterus, which may turn out to be very helpful for multi-system conditions, but immediately abortion law is going to become a big factor. And so we need to recognize kind of the dual uses of these things and the way they fit into other aspects of our law, while we try to anticipate creating a coordination as opposed to harmonization of global law. Thanks very much.
Thank you so much, both of you, for this very comprehensible and very large view that you gave us of the issues on one side and the possibilities of governance on the other side. I think that we have already several questions. I'm going to start with one who is coming from Didier Courneuil, who is from the transhumanist French movement and is questioning the Oviedo Convention. What is, and challenging this Oviedo Convention, wondering why, if it could be possible to have healthier life, would it be not allowed to make that available for our descendants? Eric, you want to comment first? Eric, you have to activate your mic.
It's a great question. I think Alta alluded to it as well, and she may want to comment on the convention. This is not one of those, yes, you're right, no, you're wrong questions. Clearly, the perspective that has been guiding not just genomics, but research involving human participants for decades has been that challenge of risk and benefit. What are the opportunities that can enhance well-being, where the word enhance becomes a somewhat loaded term, enhance to make someone beyond the capacity of normal human functioning is what gives people a pause. But the idea of being able to allow science to help, to heal, and to prevent harm are principles that I think are included in that list that I provided at the end of my presentation. The devil obviously is in the details of what would be permitted. I will want to say one more thing, because this may sound like a bit of an ambiguous answer. Most of what we talk about is responsibility within the context of rational deliberation by governments and society. And I'm perfectly content to live in a world where there are some societies who wish to be more progressive in their pursuit of science to help their community, and those that may be more modest in their pursuit. That is within a large universe of rational deliberation. Where it becomes concerning are those who are at the sort of tail end of that curve, who intentionally wish to use technology, not simply to help human well-being, but to take advantage of the power of technology over humanity. And that's where, of course, the worrisome risks of not only narcissistic behavior, but overt militaristic use of technology raises those concerns. The flip side at the other end of the curve are those who are Luddites with respect to the capacity of science and technology to help and to heal.
Thank you, Eric. Alta, you want to have some comments on this?
I would, but actually I think it's more important to let other people speak. So let's keep going.
Okay.
There's another question.
So, Alta, maybe some precision or remark Françoise Baylis is making on YouTube, because you said that you spoke of certain US regulation. She's wondering about the US Further Consolidated Appropriation Act 2020. Right. Omnibus spending bill contains one provision that restricts federal funding of human embryo research. And she's mentioning also the US FDA that cannot consider applications for clinical trials involving heritable human genome editing. So it seems that maybe you should be just a precision on the US law,
sure.
or what you said.
Yeah, and as I said, I thought that her characterization of the US as having this germline editing illegal was slightly misleading, because it's not germline editing per se that's illegal. It is a related issue, which is the spending of money on germline editing. That's the budget bill that she mentioned, that the FDA is not allowed to spend its time, because that represents money, on reviewing an application to begin a clinical trial. those budget appropriations bills go through a process every 2 years. So it has to be renewed every 2 years for that particular restriction to stay in place. Whether it'll be removed or not, I don't know. Until it's removed, you cannot pursue germline editing legally, but it's not the germline editing that is the issue so much as it is the funding, the budgeting. On the other, the embryo research, that's a long-standing restriction on the use of federal money to do research that uses human embryos. You can absolutely do research on human embryos using private money or money from about 45 of the 50 states that don't have any restriction on that. So as I was saying, the US is peculiar because the federal system of limited federal powers means we often indirectly affect things without actually saying that they're legal or illegal. And it just makes it hard to explain and probably not all that interesting for most people around the world.
No, it's something that we could continue and illustrate with the COVID crisis, because, for example, Pfizer is explaining the high price of the vaccine because they didn't want to use the federal funding. And
exactly.
Whereas others are explaining they will make the vaccine very cheap because they use federal funding. I have a question, maybe you could continue, of Emmanuel Sebag. Is it realistic to aim at fully harmonized policies? All technology rely on local policies. So what is different with gene editing, and which topics do we want to agree at on a global level?
Eric?
Me or Alta? How about I start? Because I think I was the one who used harmonized. And at the moment I said it, I realized I probably should have put it in quotation marks. And I rather like Alta's term of coordinated. Harmonized, not to push the musical metaphor, raises a few issues because it suggests that there is literally one level of agreement that can occur. At least that's the perspective that some might have. I'm rather open to the idea of as I alluded to earlier, I don't know if I would call it local in response to Emmanuel's comment. And hello, Emmanuel, glad to know that you're online. But rather, how do we accommodate the issues and interests of jurisdictions that have every right to decide how permissive or restrictive they wish to be? What I would like to see, if it were, in my power, is to have everyone participate in this discussion, to have every jurisdiction have a say in how they wish to either regulate, permit, promote, encourage, or even prohibit an actual technology. Where it becomes challenging, of course, is that the world is not made up of geographic maps where technology ends at one border and will never leap over that particular fence into another country or state or province's backyard. So harmonization, which I know is a technical term in law and policy, is more of a philosophic concept that I was suggesting. So do I agree that an individual state may wish to limit? Absolutely, they should. There will be consequences for any decision that a jurisdiction takes, whether it's permissive or restrictive.
Thank you. Alta, just a word. We have still 2 minutes.
So I'm going to be very brief. I think that we should celebrate the fact that the world is filled with countries that have very diverse cultures that balance risk and benefit very differently. Some that would say that any possible risk to the future should be avoided, even if it means we sacrifice today. And others who say, I want to see everybody today live as well as they can, and I'm optimistic that technology will improve to handle problems if they occur in the future. These are legitimate value differences. We have countries that are pro-natalist and countries that are anti-natalist. These are legitimate value differences. And the only way that we can actually experience all ways of living is to, in fact, celebrate the fact that you can go different places and live different ways. I look for coordination so that we don't obstruct one another and that we don't export injury to one another, but at the same time, not expect that everybody be the same.
Yes, and thank you so much, because if we want to have this multicultural view and allow each of these views to have the floor and the place, we need organizations such as ARRIGE, where the dialogue and where the variant views can be expressed, and coordinated in some aspect. And it's really one of the fundamental mission of ARRIGE to foster this dialogue and to continue the discussion, as you said, Eric, that it was just a milestone. And with ALTA, we will have 5 principles on the way to view human genome editing governance. And we hope we will continue this conversation because obviously it's a dynamic process, and ARRIGE will be here in January, but also next year to continue the discussion. And so now we let the floor to Jennifer and Marion for an incredible wrap-up session, because so much was said all during this afternoon that I already congratulate Marion and Jennifer for their capacity to make a wrap-up of all these so rich sessions. Thank you so much, all of you, to be here. Thank you, Eric. Thank you, ALTA, for this great session.
Hello, everyone. So we're going to be very brief because we're already out of time. Thank you so much for joining us during a very intense Saturday morning, afternoon, or evening, depending on where you are on the planet. And I must say the COVID-19 pandemic, you know, considered as one of the most crucial global health calamity of the century, caused obviously many issues throughout the world, but causing this meeting to happen online rather than being held in Spain, I must say, was not one of the negative impact of the COVID-19 pandemic. So as Martina Crispo raised, there are more and more complex issues as technology keep developing at a very fast pace. And we can see that the genome editing technology can be used to some extent to face the challenges raised by the COVID-19 pandemic. It can also be used to end malaria, as Elena Gomez-Diaz raised, can be used in agriculture, as Ruud de Maat developed, and finally can be used in human, as you already know, and it triggered so many reports and articles will not develop on that, as said Eric Melin. But the question is, now that we said that, what do we do now? As Altasharo pointed out, regulations are not uniform throughout the world, and probably speaking about coordination rather than you know, uniformity or harmonization, as she wrote very recently on the chat, is a good way to tackle the issue. One other issue we need to address now in concrete ways are the inclusion of patients and lay public in the debate. And I think it was very important to have a patient organization perspective, thanks to Nick Mead today, to, you know, address this issue, and we'll never repeat enough how important it is, and to provide a layer of education before, you know, holding those kind of debates in a more inclusive way. And definitely ARRIGE faces some challenge and has the duty to act in that respect. And finally, there are so many guidelines, reports, expert groups, etc., as Eric Melin said, despite his optimistic gene. And this is true, and it is difficult probably to, you know, isolate the key takeaway and to adjust and adapt as technology evolves and society evolves as well. But clearly, starting with multidisciplinary exchanges is key in this process. And if I can quote again Altasharo, we cannot make good policy without good facts. So thank you for being part of the exchanges today. This is not the end, this is only the beginning. So feel free to continue the discussion on Slack, by emails, by any vector possible, and be assured that ARRIGE will be there. I leave the floor to Jennifer.
Thank you, Marion, and thank you for the great summary. I'm going to be shorter than you, and I'm simply going to begin by thanking all of you who participated today in this 4-hour intense, as Marion said, discussion. I also want to thank the members of the ARRIGE board who put this meeting together. I played a very little technical role in all of this, so I really wanted to take my hat off to those of you, and you know who you are, who were extremely active. And last but not least, I want to thank our speakers that we have had ever since 2 o'clock, 3 o'clock this afternoon. Each talk was extremely professional, well presented, just in time. I haven't attended a Zoom conference as smooth and as rich as the one that we had this afternoon. So thank you to the speakers. It's really exhilarating to know that we are all speaking to each other from various places in the world. And this is something that the COVID has done has had a sort of a positive impact, even though we're all zoomed out, all of us. This has allowed for this meeting, the COVID has allowed for this meeting to occur with so many people from all over the world at, of course, a reduced cost and no carbon imprint. So those will be my last words. And I will give the floor back to Luis, the president of ARRIGE. And like Marion says, we are here 24/7. Bye.
Thank you, Marion and Jennifer. That was a nice wrapping up. And I know that Marion has been not only taking notes, but also making a very excellent drawings that she might be sharing later on. So you would be amazed to see this. So thanks to all the speakers. I think we all learn, and I'm just, my head is boiling with thoughts. And now I need to go back and reflect. I mean, this afternoon with all these 4 different talks that were excellent as well before the break. Again, apologies for not being able to broadcast properly the Gaetan Burgios video, but don't worry, we will upload it at the ARRIGE YouTube channel. And the same, this meeting will also be uploaded publicly and will be shared. So thanks again, and we will continue discussing, as Marion pointed out, in many different communication channels. Thank you and stay safe.